This instructional video is part of the tutorial series for the Infinity Analyze microscopy software. A new cell counting function has been added to the package with release 6.0. In this session, we'll show how to use this function and take a look at a couple of other small features added to Analyze 6.0. The steps that will be covered in this session include setting the cell counting options, interactively identifying the target areas, and reviewing the results from that operation. The cell counting function is based on the photometric pixel response measured by the camera. That is to say that if the cells or areas of interest can be identified exclusively through a distinctive spectral signature in the red, green, and blue pixels, then the cell counting operation will be able to identify the areas based on the operator's selections. On the screen is a live preview window showing a sample under the microscope. Before we capture an image, I'm just going to reference the zoom slider, which is new in Analyze 6.0, so we can zoom the live preview window in and out. Also, as part of this session, I want to look at Help Settings, where we've added a new interface dialog. You'll notice at the bottom of the screen that we have the help desk enabled, as well as the measurement, histogram, and photometry auxiliary windows. These can now be turned on or off dynamically using the tab and the settings menu. We'll proceed by capturing an image of the sample shown on screen. With the capture image displayed, we'll now take a look at the cell counting options. These are accessed through the measure menu by selecting cell count options. The cell counting options can be configured to provide the best results based on the image contents. If the areas of interest all have a similar response in the red pixels, for example, then a low threshold will be required for the red channel. At the same time, if the green and blue pixels for the same areas of interest have a wide range of intensities, then selecting a larger threshold for these colors will mean that the classification process will take the operator fewer mouse clicks to complete. If the thresholds are set too small, then multiple mouse click operations will be required to identify the areas of interest. While setting thresholds that are too large will mean that unwanted regions of pixels will be included in the cell counting. It will take some experimentation to find the threshold settings that work most effectively for performing the cell counting function on your own samples. In this sample, we've got tolerance for the red set to 22 pixel intensity counts, green is set to 25, blue is set to 15. On the left-hand side, we have colors to choose for identifying the nucleus. As the operator clicks on the mouse, a red shaded mask will appear when we have the nucleus set to a red color. On completion of the cell counting, the boundary will be shown by a yellow outline, and the index numbering will appear in blue. You have choice over font size for the labeling, and a closing parameter. The closing parameter defines the tolerance for holes or gaps within the areas identified, and at the same time defines the space between adjacent regions that will also be closed if they are less than this specified distance in pixels. Now we'll proceed on to the cell counting operation. Going to the measure menu and choosing cell count. The first thing you notice is that the cell count help desk gives an explanation of the steps required to complete a cell counting operation. There are multiple combinations of mouse clicks, and at least for the first few times you're using cell counting, it's a good idea to keep the cell count help desk auxiliary window open. I'm going to go back to the zoom slider and zoom in on my sample so we can see more clearly what's going on. You'll notice the eyedropper icon, which is showing up for the mouse pointer location. Each time the operator clicks the left mouse button, the intensity of the pixels immediately beneath the area they've clicked is analyzed, and all similarly classified pixels throughout the scene will also be identified and covered by the red mask. The operator would then move around and select similar pixels that are as yet unclassified in the areas of interest. I'll zoom in a little further on the sample so we can see a little more closely. You can continue to identify pixels within your area of interest until your cells or regions have mostly been identified. If you carry on aggressively and identify too many pixels, for example, if I begin to identify some out here near the periphery, I'm going to suddenly get pixels that are uniformly identified throughout. Edit Undo will take us back a step. You can also limit the cell classification to a single area of interest by holding down the Control key when you left mouse click. Holding down the Control key while you perform a right mouse click allows you to identify areas that have been joined together. For example, in this case, were these a little closer together, they would have been joined together. If I hold down the Control key and do a right mouse click, it will minimize the area on those cells. Also, if I'm, as mentioned, holding down the Control key when I left mouse click, it limits the classification to one nucleus or one cell of interest. So that gives a quick overview of how the cell identification would take place. As soon as you're satisfied with the overall classification that you're seeing, you can double right mouse click in any area that's not covered by the red mask, and the cell counting operation is immediately performed. You'll notice in the measurement auxiliary window a total cell count of 641 identified cells has been shown in the measurement results, and because I was calibrated for my 40x objective, the total area of classified cells has been identified. Additionally, over here in the photometry auxiliary window, I have for the red, green, and blue pixels an average pixel intensity, the minimum, maximum, and standard deviation. As with the measurement window, as we've shown in other tutorials, you can highlight areas in the photometry window and highlight them with the mouse and then click on them. If you have an Excel spreadsheet open in your tool tray and you drag your mouse over it, it will open the Excel spreadsheet, and you can release the mouse when you're in the Excel spreadsheet and can now perform simple operations on the spreadsheet for your results. Finally, if you return to the Measure Cell Count menu, you get your mask layer back, and you can continue to add or remove or edit your mask layer before double right mouse clicking to perform the operation once again. And lastly, to get rid of the measurements, as with all of measurement functions, you can use the Clear Measurements option to get rid of those measurements. That concludes the video tutorial on cell counting. Be sure to investigate other video tutorials in the Infinity Analyze series.